All right, class, so let's get on to our final lecture of the semester, part 2 of chapter 16, Integration of Metabolism. Again, here's our chapter theme from Andrew Hellstrap and Michael Newton. Thus, as Christians, we study biochemistry knowing that Christ ultimately stands behind the ordered mechanisms of life. And I think we've all seen the ordered mechanisms of life, in particular in the previous video lecture of chapter 16. A consistent God upholding a consistent universe whose resources we can harness for the good of mankind. Well, let's just go ahead and jump into some content, okay? So each organ in the mammalian body, remember our bodies are composed of organs, contributes to the individual's function in several ways. So for instance, organs can consume energy or also supply energy-rich nutrients. Signal molecules offer an important control mechanism for integrating these processes, and we've talked about that in the previous video lecture with the signal transduction cascades at the receptor level. And also nutrient transport across cell plasma membranes is an important feature of organs. Okay, so make sure you understand this and that you understand what organs are, how tissues and cells are involved in making organs, but organs or the overall system that achieves function in your body. Okay, so when you think of organs, spleen, pancreas, your liver, stuff like that. Okay, so if you want to look more into organs, I'm sure you can research it on the internet. But for this class, we're going to take a deeper look at what these certain organs do, in particular the liver and the pancreas. So despite their constant requirements for energy, mammals like ourselves only consume food intermittently, meaning we're not always needing food for energy, but we're able to eat some food and then we can chill but have the energy derived from it, and then we can go a period of time without eating food but still have energy. And this is based on what's called the fasting state and the fed state, okay? And we're going to talk about those in detail. This is possible, meaning intermittent eating, because of the elaborate mechanism for storing and mobilizing energy-rich molecules derived from food. So remember, plants, autotrophs, they derive their energy source from the sun in the form of photons, and it undergoes photosynthesis. And then we as heterotrophs end up eating these plants and eating animals that eat the plants. And what happens is our bodies are able to extract energy from those chemical sources, and that's how we get our energy, not necessarily from the sun directly, but from other things that eat other things that is eventually derived from plants that harness its energy from the sun. Moving forward, I want to talk to you a little bit about these metabolic pathways that you've already seen before, so don't let this slide intimidate you or scare you. But animals must have metabolic integration and the regulatory influence of hormones. We talked in detail about how hormones and regulation is involved in metabolism in the last video lecture. But I just wanted to give you an overview of what all we've learned in this class. So if you start with the left-hand side of this drawing, the upper left-hand corner, you can kind of see glucose. It's kind of small, but glucose can go to G6P, glucose 6-phosphate, and it can undergo glycogen storage, which is called glycogenesis or glycogen degradation, glycogenolysis, to arrive back at G6P. And G6P can then be shuttled into the pentose phosphate pathway, or it can proceed forward in glycolysis to fructose 6-phosphate and then fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate can be split into glyceraldehyde 3-phosphate, G3P, or dihydroxyacetone phosphate, DHAP, which DHAP can be involved in fatty acid synthesis, lipogenesis by being converted to glycerol phosphate and then triacylglycerol, and then mix with this acyl-CoA, you can get fatty acids. Or DHAP, dihydroxyacetone phosphate, can be converted to G3P, which proceeds forward with glycolysis, giving you glycerate 1,3-bisphosphate, glycerate 3-phosphate, glycerate 2-phosphate, and then phosphoenol pyruvate, which then yields pyruvate. And of course, this doesn't showcase gluconeogenesis either, where pyruvate can then be converted to oxaloacetate and then phosphoenol pyruvate, and then proceed forward backwards back to glucose. And so you see how the integration of all this is a key feature of metabolism. Substrate concentrations are also an important factor in metabolism. So depending on how much substrate you have, it's going to depend on whether it needs to be made or if that process needs to be halted. Yeah, so I'm going to go ahead and introduce to you the postprandial state and the post-absorptive state. which is essentially the fasting fed versus fed state. I kind of got those backwards. The postprandial is your just after a meal, so that would be your fed state, and the post-absorptive state is going to be your fasting state, meaning either periods of prolonged fasting, or maybe you just slept for the night and you didn't eat for several hours. And so, again, this is another intimidating drawing. I don't expect you to memorize every single thing. I think I've already tested you on most of this, okay? But why don't you go to the top middle of the drawing where you see phosphoenol pyruvate and how that can be converted to pyruvate via glycolysis. And pyruvate, of course, can be converted to lactate, especially in anaerobic conditions. But notice that I had mentioned previously in this course that certain amino acids can yield pyruvate. And you see those 6 right there: alanine, cysteine, threonine, serine, glycine, and tryptophan. And so breakdown of proteins can yield pyruvate as well. Pyruvate then can be converted to acetyl-CoA using the pyruvate dehydrogenase complex. And of course, acetyl-CoA can undergo the citric acid cycle, and alpha-ketoglutarate can be converted to various amino acids as well, which is the third intermediate of the citric acid cycle. As you proceed forward, you may notice that succinyl-CoA can be formed from amino acids as well: valine, methionine, and isoleucine. And of course, fumarate can be synthesized from phenylalanine and tyrosine. So do you see the integration in all this? Even if you go up and look at oxaloacetate, the final intermediate of the citric acid cycle, you see how that can be converted back to phosphoenol pyruvate and gluconeogenesis, as well as be converted to aspartate, which can be converted to asparagine, and so on and so forth. You see their urea cycle off to the left-hand side. And so the point is that there is all kinds of nutrient metabolisms going on in the fasting feeding cycles. And it's so interesting to know that proteins can be used in energy metabolism by converting to pyruvate or something of that sort, and also entering into the citric acid cycle and all kinds of things. So I hope that this overview of what all we've gone over is just making you marvel at how God has created biochemistry of our bodies. The feeding phase, so this is going to be the fed state when insulin is high, right after you ate a meal. So the feeding phase involves food movement. Okay, so obviously you just chewed your food mechanically, you swallowed it, and the food needs to be moved, and then it's digested and then absorbed into the blood and lymph in your GI tract, the gastrointestinal tract. Your smooth muscle contraction propels food along the tract. So if you're having trouble understanding, you have smooth muscle, skeletal muscle, and cardiac muscle. And of course, smooth muscle is going to be involuntary movements that you're not conscious of it actually going on, but what happens is in your intestines, it moves food through smooth muscle contraction. And then also in the early postprandial state, sugars and amino acids are absorbed and transported in the portal blood to the liver. Okay, so again, you just ate, you just mechanically chewed your food, you digest it, and your smooth muscle starts moving it along. And again, digestion, it's broken down. And what happens is it's absorbed, and we're not going to get too much into it, but you may recall for instance, like in lipids, the enterocyte cells and all that. But nonetheless, it gets into your blood and gets transported to your liver. And your liver is such an important organ. We've talked about it a number of times. And so take a look at this drawing. You're going to see it a couple of times in this slideshow. But there's your liver there kind of in the middle of the drawing. It's the focal point, if you will. And if you look, you can see your GI tract, your small intestine there. Kind of looks like a spaghetti noodle or something like that. And your food is traveling through that through smooth muscle contraction. And then what happens is the nutrients and all the components of your food get absorbed into your portal blood through various ways. And then it gets sent to the liver, in which liver processes glucose, amino acids, and then glucose can then be sent off to your brain because glucose is the primary fuel source for your brain. Glucose can be sent off to other tissues for glycolysis or be stored as glycogen. Glycogen storage, it's the storage form of glucose. It's a polysaccharide. Or can be converted to amino acids, and then for protein synthesis. Your liver is involved in all that. Or what could have happened is some of the fatty acids, for instance, your TGs could be sent to the liver. Sorry, not the liver. It can go to adipose tissue through lymph vessels. You see the adipose tissues at the bottom right-hand corner of this cartoon, which can then be delivered to the liver and then be sent also to the skeletal muscles as you need it, okay, and also stored as glycogen, and so on and so forth. And again, you see the liver as it secretes insulin as a hormone to the liver to engage in the various processes involved in this fed state. So again, I'll let you know very clearly what I intend for you to know from this drawing, but I'm just trying to get you interested or whet your palate for what all is going on in biochemistry. I don't want you to get overwhelmed to where you give up, but I do want you to marvel at how this is going on without you even being conscious of it. Most lipid molecules are transported in the lymph. Okay, so lipid molecules, you already know this from chapter 12, lipids, but most lipid molecules are transported in the lymph as chylomicrons. Okay, remember chylomicrons are lipoproteins composed of triglycerides or triacylglycerols and embedded apolipoproteins and some phospholipids and some cholesterol, all kinds of stuff. And it's a transport molecule, okay, and it sends all those molecules elsewhere in the body. So this is going to be the dietary transport molecule as opposed to VLDLs and LDLs and IDLs and all that type of all those lipoproteins. Chylomicrons then pass to the bloodstream. You know, I told it traveled through the lymph. It goes into the bloodstream, and it ends up providing TGs, triacylglycerol, to the muscle or skeletal muscle and adipose tissue for fat storage. Then the chylomicron remnants, which are left over, then deliver. So chylomicron remnants, recall that your chylomicron was then plucked apart by lipoprotein lipases, leaving a chylomicron remnant, which I just told you at the time it was processed by the liver. These chylomicron remnants they then deliver phospholipids, cholesterol, and a few remaining TG molecules to the liver. So I told you the chylomicron remnant went to the liver. I didn't really tell you that much about it after that. So what it does is after all those TGs are plucked off of it, it's left with phospholipids and cholesterol, and a few remaining TG molecules that gets processed by the liver, and with the and actually, I'm going to go to the next point. The phospholipids, lipids, and the proteins are then incorporated into lipoproteins for export in the liver. Okay, so what I'm trying to say is this. Chylomicrons, which are your transport molecule from dietary sources, pretty much get all your dietary sources in one compact little ball, if you will. It travels through your lymph, and then it arrives eventually in the blood stream, which then goes to the liver, but it does get picked apart. So the TGs, for the most part, get picked apart by lipoprotein lipases, leaving you with chylomicron remnants, which pretty much consist of phospholipids, cholesterol, and a few TG molecules. These molecules are processed by the liver, but what happens in the liver is that phospholipids, lipids, and proteins are incorporated into lipoproteins and exported to other tissues as needed. Okay, so chylomicrons go to one particular place, and then the liver repackages the other materials and sends them off to other parts of the body. Now, I skipped that middle bullet point. So cholesterol is used to make bile acids, and fatty acids are used to make phospholipids, just to reiterate what the purpose of these things are, what pathways they may undergo. Now, the question of this slide is, where does insulin come from? Okay, so glucose movement from the small intestine to the liver stimulates beta cells in the pancreas to release insulin. So when you think pancreas, think insulin secretion. Now, beta cells are specialized cells located in the pancreas that are simply insulin producing cells. And so glucose movement from the small intestine to the liver, so you just ate, there's a lot of glucose there in your small intestine, gets absorbed, travels, makes it all the way to the liver. Now your liver has a lot of glucose, your body sends signals, it stimulates beta cells in the pancreas to release insulin. And what insulin ends up doing is it triggers glucose uptake, glycogenesis, meaning synthesis of glycogen, the storage form of glucose, fat synthesis, so you're going to make triacylglycerols, and also storage, and generally stimulates protein synthesis. And so insulin triggers a lot of things: glucose uptake, glycogenesis, fat synthesis and storage of fats in adipose tissue, and it also stimulates protein synthesis for the most part, okay? And glucose uptake, of course, is going to be involved in like glycolysis for energy. So yeah, you eat food, you want energy, and so glycolysis is also stimulated. The fasting phase, aka the post-absorptive state, this is what's going on. Glucagon is released, so glucagon is kind of like insulin's equal opposite. So whereas insulin is in the fed state, and it makes your body undergo protein synthesis, glycolysis, and not gluconeogenesis, glycogenesis, the synthesis of glycogen, um, and also fat synthesis, lipogenesis, and then fat storage. The fasting phase, the post-absorptive state, glucagon is released as glucose and insulin levels fall back to normal. So you're done eating, your glucose levels are starting to deplete back to normal levels, your insulin levels start falling back to normal, and so now glucagon is released. This is your fasting phase. What glucagon does is it prevents hypoglycemia, which is low blood sugar, by stimulating glycogenolysis and gluconeogenesis in the liver. Okay, let me unpack what I just told you. So you're not eating anymore. You have fasted for quite a while, and your levels of insulin and your levels of blood glucose start to fall a little bit back to normal. And so glucagon is released, a peptide hormone which stimulates glycogenolysis. Why would it do that? Well, you don't have a supply of glucose anymore. So what that does is it starts breaking down glycogen for glucose usage. Okay, so what you're doing is you're withdrawing money from the bank that was storing all your money. Okay, because you need a little bit more. And it also starts gluconeogenesis, which takes pyruvate and other molecules and sends it back up glycolysis in the reverse way to generate glucose for other processes. So essentially, all what you're doing is you're harnessing other pathways to stimulate glucose synthesis rather than getting glucose directly from your food itself. So if the fast becomes prolonged, meaning it's not just a couple hours after you ate, but you've actually undergone, let's say, sleeping in your hours without food, maintenance of blood glucose levels occur by fatty acid mobilization. And so let's take a look at two different things. For instance, what you're going to look at is your brain versus muscle. Okay, so your muscle is going to go to fatty acid mobilization for its primary fuel source. And so normally glucose would be a primary fuel source, but what happens is that fats can get broken down through lipolysis to generate the fatty acids, and then beta oxidation can then yield a lot of energy, as you saw in chapter 12 concerning lipids. Your brain needs glucose for energy, okay? It's the primary fuel source. So whenever you start using fatty acids for your fuel source in your muscles, it opens up those glucose levels to go straight to your brain because your brain is an energy-hungry organ and uses a lot of glucose as its primary fuel source. In fact, it lacks fuel storage capabilities and uses about 120 grams of glucose daily, which is about 420 kcals. Um, on your student notes, I do want to point out that you had the wrong drawing in this particular slide. It says figure 1614, the post-absorptive state, but gives you the postprandial state in the cartoon. So make sure you look at this slide for the correct drawing. And so as you can see on the right-hand side, the pancreas secretes glucagon and not insulin, and then it goes through the portal blood, and then it stimulates a number of other processes. You notice that glucose is stimulated, and then it ends up traveling to the brain, and amino acids can yield glucose from protein, so on and so forth, all this occurring in your liver. And anyways, you see in your skeletal muscle, fatty acids can go and be harnessed. I'm looking at the bottom right-hand corner because your adipose tissue has glycerol and fatty acids, but those fatty acids can then be used in your skeletal muscle for energy, and also amino acids can be broken down into from proteins. So protein breakdown yields amino acids as well, and glutamine can be secreted into the small intestine and traveled. So, and also alanine can be sent to the liver, and alanine can generate glucose also through gluconeogenesis by being converted to pyruvate. And so hopefully a lot of these processes look familiar, so make sure you pause this video and study this drawing a little bit. And like always, I'll do my best to make very clear what I expect you to know about all this discussed in this slide. So during extraordinarily long fasting periods, aka starvation, so now we're not talking just overnight or something like that, but maybe 24, 36, 48 hours, we're talking a long time of not eating. What that does is it leads to metabolic changes to make sure you stay alive, okay? Because your brain needs glucose, and if you run out of glucose, you're in trouble. And so how how do you maintain a healthy, normal metabolism and survive whenever you go through long periods of starvation? What happens is additional fatty acids from adipose tissue, meaning it breaks down the TGs into fatty acids and glycerol, and those fatty acids are then they are sent off to undergo beta oxidation in the mitochondria to form ketone bodies. Okay, ketone bodies from the liver are then mobilized. So what happens is, remember, ketone bodies are formed when excess acetyl-CoA from beta oxidation of fatty acids in a mitochondrial matrix of liver cells is converted to ketone bodies such as acetone, acetoacetate, and beta-hydroxybutyrate. These ketone bodies can then be used for energy. So in extreme periods of starvation, instead of just forming acetyl-CoA and generating the normal amounts of energy through the citric acid cycle and electron transport chain, it actually doesn't go to acetyl-CoA, but rather all the way to ketone bodies, and then these ketone bodies are going to be used for energy in these extreme periods of starvation. So keep in mind that glycogen, though it is a storage form of glucose, it's depleted only after several hours. So gluconeogenesis plays an important role in energy during the fasting state. So large amounts of amino acids from muscle protein are also used for this purpose. So you can use amino acids derived from protein degradation, and it can eventually be converted to pyruvate. Remember those 6 amino acids I discussed earlier? And those can be used once it is formed as pyruvate. Pyruvate can undergo gluconeogenesis to reform glucose for energy. And just to reiterate, those 6 amino acids were alanine, cysteine, glycine, serine, threonine, and tryptophan, but most notably alanine. And I do want to let you know that after several weeks, like I don't know if any of y'all have ever fasted or undergone any long periods of starvation, but your body starts undergoing a lot of changes. You have headaches, you feel very tired, you get very cranky. But after a couple days, you get used to it. Not to where you're not hungry, not to where you don't crave food, but your body's just more adapted to it. So it's finished undergoing all those physiological changes, and now it realizes this is the new normal. Okay, so after several weeks, the brain can actually become adapted to, intermittent fasting or periods of starvation, and it gets used to using ketone bodies as an energy source. So here's a slide on feeding behavior. Regulating feeding behavior involves hormone and neuronal signals. Remember, neurons are brain cells, as well as sensory input from the environment, aka the 5 senses. And so, as you look in this slide, you'll notice that we get hungry or we feel, well fed and not hungry based on the brain. Okay, based on the brain. And what happens is not only does the brain send neuronal signals and also hormonal signals, there's also your 5 senses. Have you ever gotten hungry when you smell something, or you kind of lose your appetite whenever you smell or see something as well? Or if you taste something and it's not appetizing, or you taste something and it tastes better than you thought, and then you just go to town. And so the 5 senses also play a role in your feeding behavior. Both are integrated in the appetite. Both are integrated in the brain to regulate appetite. And remember, the hypothalamus is involved with this behavior. And we talked about the hypothalamus in the last lecture video in this chapter. And lastly, our last content slide of this semester, so congratulations. I just wanted to give you a brief sketch on diabetes. Okay, so we've all heard about diabetes and how there's type 1 and type 2. Now that you've gone through this class, hopefully you can understand what each one is. So type 1 diabetes is caused by the destruction of insulin-producing beta cells by the immune system. So your immune system attacks them, and then they're destroyed, okay, or there's some destruction, and so it's unable to produce insulin. Symptoms usually occur abruptly, with the most serious symptom being ketoacidosis. If you don't know what that is, I encourage you to go look up what ketoacidosis is. The way you treat type 1 diabetes is you use insulin by injection, because if your body's not producing the insulin needed, you're going to need to get it from some other source, and you're always having to monitor your blood glucose levels, because if it drops too low or gets too high, you're going to want to just make sure you keep track of that. And so insulin, of course, stimulates glucose movement, glucose storage into glycogen, and also fatty acid synthesis. And so if you don't have insulin, it's going to cause a lot of problems, okay? Type 2 diabetes is caused by insulin resistance, remember, due to downregulation of insulin receptors. And when you're obese, you're typically more at risk. Okay, downregulation just being, you have these insulin receptors on cells, and you may recall that these types of receptors for insulin are receptor tyrosine kinases of that particular family of receptors. But anyways, your target cells have a certain number of receptors for insulin. But if you have too much insulin being produced because you're eating too much over long periods of time, what happens is there's actually this downregulation, which is a desensitization effect, where there's less receptors on the cell to be affected by insulin. And so that is what's going on in type 2. But good treatments for type 2 diabetes would be diet and exercise. A good, healthy, maintained diet and good exercise. I already presented your homework in the previous lecture video, so make sure to go look at that. But here they are again, into chapter problems 1, 13, 24, 26, 27, and 58. So make sure you get those finished by the due date. And to finish off this chapter, I hope that you can agree with Chris Tomlin and the other people that have sang this song, "The splendor of the King, clothed in majesty, let all the earth rejoice." And how great is our God. So I hope you have gotten close to God during this class. I hope that you have been challenged in this class, and I hope that you're satisfied with the education you received from this class. I hope y'all all have a blessed time, and I've really enjoyed teaching y'all this semester.